organizers for inviting me to present our research today. My name, as was presented, is Fabienne Meier Abt. I'm an MD PhD and currently I work at the University Hospital of Basel. But today I'm going to talk about the research that we performed at the Friedrich-Miescher Institute for Biomedical Research. And I'd like to talk in the next coming minutes about how pregnancy at early age protects against breast cancer. Can you hear me? Yeah, okay, wonderful, thank you. So first I'd like to give a quick background. It is well established that pregnancy at an early age protects against breast cancer, and this was shown in numerous epidemiological studies, and it's illustrated on the first slide. And here on the y-axis you can see the relative breast cancer risk, which always is compared to the age-matched nulliparous individuals. And here on the x-axis is the age in years. And as you can see in blue here, if a pregnancy occurs before the age of 20 years, there's a greater than 50% lifelong protective effect against breast cancer. This is shown here. Importantly, as the age of the first full-term pregnancy increases, this protective effect is lost. This is shown here in green and even more extreme in red when the first pregnancy occurs after the age of 35 years. And then there's even an increase in the lifelong risk to develop breast cancer. Now, given today's societies where the age of the first birth is delayed more and more and more, ways are warranted to mimic this effect without women having to go through teenage pregnancies. And for this, the molecular mechanism that explains this phenomenon needs to be known, and unfortunately, up until now, this was not really understood. Now, fortunately for us, in rodents, there is even a greater protective effect of early age pregnancy. It's greater than 75%. And this means that rodents can be used as a model system to study the mechanisms that underlie this phenomenon. So several mechanisms have been, or hypothetical mechanisms have been put forward that can potentially explain the protective effect of early age pregnancy. They include a change in the proliferation differentiation potential of specific mammary epithelial cells. They include systemic changes in circulating hormones and changes in the stromal composition of the mammary gland. Now, importantly, these different hypotheses are not mutually conclusive and exclusive, and it's likely that several of them work together to bring about the full effect. So, early age pregnancy protects against breast cancer, but the mechanism is not known. And on the basis of that, we asked the question, what is the effect of an early age pregnancy on the gene expression profile and on the proliferation and differentiation potential of the different mouse mammary epithelial cell subpopulations, since these are the cells that give rise to breast cancer. But before I'll go into the details of our research, I'd like to give a very quick overview over the memory cell type hierarchy. It has been introduced here before, but just a very quick review of it. So here you can see a breast with acinar structures and ductal structures. And here on the right is a cross-section through one of the acini. So in green, you can see the luminal compartment, which is responsible for milk production and secretion. And in red, you can see the basal compartment, which is responsible for milk ejection. Together, they form the mammary epithelium, which is surrounded by a basement membrane, which separates it from the stromal cells, here shown in brown. And here you can see cross-section through the mammary epithelium. So again, in green is the luminal compartment with differentiated luminal cells and differentiated luminal hormone sensing cells, which are positive for estrogen receptor and progesterone receptor, but there's also luminal progenitor cells. And in red, you can see the basal compartment, which contains differentiated myoepithelial cells, but also basal stem cells and basal progenitor cells. Here on the right, there are the markers, the biomarkers, which can be used to differentiate between the different cell types, and also importantly to isolate them. They're here shown for mice. So in our research, we isolated these different mammary epithelial cell subpopulation using FAC sorting and the markers that I've just shown you. We then performed transcriptome and bioinformatic transcription factor analysis, which was followed by pathway analysis using Ingenuity and gene set enrichment analysis. We performed functional analysis using colony formation assays and mammary gland reconstitution assays. We also did immunohistochemistry and rescue experiments with recombinant proteins. This leads me to before I'm coming to the results section actually, I'd like to very quickly explain the early age parturition protocol that we used. So we mimicked the early age pregnancy scenario in humans by mating mice when they were 42 days old. That's very young for mice, and this time point was chosen to really mimic this early age pregnancy scenario. 21 days were allocated until parturition, another 21 days were allocated for lactation, and 40 days were allocated for mammary gland involution before the cells were harvested. All Paris mice that had undergone early age pregnancy were compared to age-matched virgin control mice. And this now leads me to the results section. So first, the transcriptome analysis in mammary epithelial cell subpopulations, both from Paris mice and from age-matched control virgin mice. So as you can see here, there were changes in gene expression in all epithelial cell subpopulations, but not in the struma. And by far the greatest number of genes with changed expression was observed for the basal stem and progenitor cell subpopulation. This is shown over here. And when looking at the exact nature of the genes which changed their expression, we found a strong upregulation of differentiation genes, here shown in blue, with the fold change on the microrange shown in brackets. And we also saw a strong downregulation of Wnt signaling, this is shown here in green, and an upregulation of Notch signaling, which is shown here in orange. So to those of us who are not so familiar with Wnt and Notch signaling, they are both important cell fate determining pathways, which in the case of Wnt signaling are linked to cell proliferation and carcinogenesis in basal stem and progenitor cells in the mammary gland, whereas Notch signaling is linked to reduced proliferation in the same basal stem and progenitor cells. So these results were validated not only by quantitative PCR, but also on the protein level, which also showed a strong downregulation of this Wnt to Notch signaling ratio. And a downregulation of the Wnt to Notch signaling ratio indicates that there might be substantial changes in the proliferation potential of these stem and progenitor cells. And in order to test that, we then performed functional analyses, and that's the next part, namely the effects of early parity on the clonogenic and proliferation potential of these basal stem and progenitor cells. So here on the right, on the right, you can see typical colonies which are grown from the different cell subpopulations. So here from myoepithelial cells, here from basal stem progenitor cells, here from luminal SCA1 positive cells, SCA1 negative here, and SCA1 positive cells. These ones are estrogen receptor positive, these ones are not. And on the left, you see the colonies which are grown from cells from virgin mice, and on the right, you see the colonies grown from cells from Paris mice. And here's the quantification. And as you can see, there is in green here is the number of colonies from cells from virgin mice, and in orange are the ones from Paris mice. And as you can see, there's a huge decrease in the number of colonies formed per well for basal stem progenitor cells. And this is consistent with a decrease in the Wnt to Notch signaling ratio in this cell subpopulation. You can also see that the colonies themselves, there was not just fewer of them, but they were a lot smaller. This is, it doesn't have a battery anymore, but you can see it over here. we then went on and did similar experiments in vivo. And for this, oh, there's another one. Wonderful. Thank you very much. So for this, we removed this epithelial bud from 3-week-old mammary glands in 3-week-old mice, and this left us with the so-called cleared fat pad. And into those cleared fat pads, we transplanted basal stem progenitor cells either from Paris mice or from mice which were virgins. And then we waited for 8 weeks and then assayed for reconstituted mammary glands. And here is the quantification of that. And again, in green, you can see the repopulation frequency for cells from basal stem progenitor cells isolated from age-matched virgins. And then here in orange, you can see the repopulating frequency from basal stem progenitor cells isolated from Paris mice. And whereas there was a very significant difference in the number of outgrowths that were greater or equal 25% of the fat pad filled, so the number of big outgrowths, the number of smaller rudimentary outgrowths did not change significantly. And this suggests that the repopulating frequency and the repopulating capacity of the basal stem progenitor cells was markedly decreased after early age pregnancy, but the cells themselves were not eliminated. So the stem cells were still there, and they had a potential to replicate to a minimal extent, but they were not able to reconstitute an entire mammary gland anymore. And again, this is very much in line with the decrease in the Wnt-to-Notch signaling ratio that we found in the cell subpopulation. It can also explain a decrease in tendency to produce breast cancers. This was then further looked at in bioinformatic technology. I will not go into the details of that, but I'm very happy to talk about it later on. We next wondered also about the mechanism that led to these changes in basal stem progenitor cells. And here we looked at the total cell suspensions. And here on the left, you see the transcriptome analysis from the cell subpopulation from 4 different groups of virgin mice, and then on the right from 4 different groups of Paris mice. And as you can see, there was a significant decrease, greater than 3-fold decrease actually, in the expression of Wnt4. Now, Wnt4 is known to be secreted in response to progesterone signaling by hormone receptor positive luminal cells. And when looking at those cells, this is shown over here, in brown you can see progesterone receptor positive cells, and in blue you can see progesterone receptor negative cells. Here on the left is a representative image from a virgin mouse, and on the right is a representative image from a mouse which has gone through early age pregnancy. And here's the quantification, and as you can see, there is a highly significant decrease in the number of progesterone receptor positive cells. And this explains why there's less Wnt4. Now, in order to establish causality, we then went on and performed rescue experiments with recombinant Wnt4. And indeed, recombinant Wnt4 was able to rescue the proliferation defect in the basal stem progenitor cells. Yeah. So this leads us to the following mechanistic model. We believe that an early age pregnancy reduces the hormone sensitivity of the mammary gland by reducing the number of luminal hormone sensing cells. This leads to a reduction in Wnt4 secretion, which in turn leads to a reduction in Wnt signaling in the stem progenitor cells. And this in turn leads to an upregulation of Notch signaling, which leads to an increase in differentiation, a decrease in the proliferation potential, and a decrease in the tumorigenic potential. Now with this, there were just 2 questions remaining, and that's the duration and the age dependency of these changes. I won't go into them in detail because of time reasons, but I'd like to mention that these changes, they lasted until the death of the mice. so until what is known as postmenopausal age for the mice. And late pregnancies, occurring when they were already 6 months old, did not lead to any of the changes. And this is very much in line with what we see in the humans, that early pregnancy protects, but late pregnancy does not. And supports that there is a similar, that these changes are responsible for the protective effect, or at least in part, and that similar processes happen in the mouse and humans. So this leads me to the summary. We've seen that early parenting leads to an induction of differentiation and a downregulation of the Wnt/Notch signaling ratio in basal stem and progenitor cells. It downregulates potentially tumorigenic biofunctions in these basal stem progenitor cells. It also decreases the in vitro and in vivo proliferation potential of this cell subpopulation, and mechanistically it works by a reduction in progesterone responsive and Wnt4 secreting luminal cells. And importantly, as I mentioned to you, these changes are of lifelong duration and they're not induced by late age pregnancy, which is fully consistent with what we're seeing in humans. So in conclusion, these changes potentially explain the breast cancer protective effect. We don't know whether they're, what part they form. There's probably other phenomena that account for it too. We don't know yet how important exactly this part is. We know it has some role, but we don't know yet what proportion of it makes out. So this remains to be studied. Also, the decrease in the Wnt/Notch signaling ratio in basal stem progenitor cells has been confirmed in humans, but in humans with all of the heterogeneity, further validations are warranted. And finally, and very importantly, the results open the door to future studies assessing whether inhibitors of the Wnt pathway might be used to mimic those early parity-induced protective effects against breast cancer. And this is also ongoing, first, of course, in mice, and potentially later on then in humans. With that, I'd like to thank my mentors for, first of all, Dr. Bentiris Alsch, but also Professor Gasser, Professor Rochlitz, Professor Schubler, and Dr. Smalley. I'd like to thank all Bentiris Alsch lab members. I'd like to thank the facilities at the Friedrich Miescher Institute and funding. And importantly, I'd like to thank all of you for your attention, and I'm happy to take any questions if we have time.
First of all, I want to congratulate you on this unbelievably fascinating study. And what you have done, have just sort of really brought a scientific basis based on epidemiological studies that they have identified pregnancy as a sort of a practically protective mechanism. And those patients that they undergo early pregnancy and early lactation, so the one that they have, experienced lesser number of, breast cancer overall. And we have known that for a while. That's a very elegant study. I want to really congratulate you on that.
Thank you very much.
And the things that right now we are observing all over the world, especially in Western countries, women are getting married much longer.
Mm-hmm.
And by the time that they get pregnant, they're really on a 30, 35, 80,
Mm-hmm.
40, that in a regular component is a very old age for pregnancy. And there are hypotheses right now that one of the reasons for increased number of breast cancer in Western countries is that concept of the fact that women are getting pregnant very late,
Mm-hmm.
which does not really help them,
Mm-hmm.
and it really removes them from that protective timeline that
Mm-hmm.
we all experience when we're younger. And of course, the fact remains that, I think following what you're saying, lactation also has a lot to do
Yeah.
with that protective measure to let the epithelial cells to really mature, to become who they are. And that really happens by pregnancy.
Yeah.
And if that pregnancy doesn't happen, they really stay immature
Yeah.
and so-called dysplastic. And that
Yeah.
can give rise to initiation and increased number of breast cancer. So very elegant study.
Thank you very much.
I'd like to talk to you after.
Yes, I'm very happy.
Are there any drugs coming online for the Wnt pathway that you could test in your mice?
There are. Unfortunately, there's just some in phase 2 trials now. There's nothing that is accepted or by the FDA and on the market, but people are really, really looking for those. There's small molecules, there's also antibodies that we can use in mice, but we're obviously also interested in using something that will get onto the market. And it's not just breast cancer that will benefit from it. I mean, obviously, colon cancer and so on. So there's really a strong push in trying to find Wnt inhibitors that can be used. But so far, they're quite far in the clinical trials, but it's not yet there.
So genes like S1, which all goes up, independently apoptotic gene.
Sorry, can you?
That's one.
That's one.
Apoptotic gene, and you show that it goes up.
Mm-hmm.
And this is particularly important in formulated breast cancer. It's highly expressed in there. Do you try to think about blocking the expression and see what happens?
Yeah, we have not done that. This is there listed because it's a target gene of Notch signaling. We actually have not gone into genetically changing the cells, but that would be a very, very interesting thing to do. I think DUS1 is one thing, but I think Notch signaling in itself. Now, what has been done, now not in relation to pregnancy, but what has been done is that just Notch signaling was changed in the stem cells, and they found that they proliferate a lot less. Now, the opposite is true for Wnt signaling. This was also looked at in a genetic system. And so if Notch signaling is upregulated, they proliferate less. If Notch signaling is downregulated, then they proliferate more. Now, in the Wnt signaling, it's exactly the opposite. And in Wnt signaling, they went in and they also had reporter mice. They could show it with that. But we, in our study, did not use genetic models. But that would be a very interesting next step to then look at it also in relation to pregnancy. Absolutely. Thank you very much. Yeah.
Any other question? Not, then I'd like to thank her for this presentation.
Okay. Thank you very much. Thank you.
Very nice.
Thank you very much.